The issue of chemicals in the environment is a story about science, politics, and humanity's relationship with the planet. My name is Chris Hughes, independent scientist, and this is the Chemical Journeys podcast. Join me as we uncover the story of chemicals in the environment through conversations with individuals working in this space. So, let's get started. Hello everyone, and welcome to the Chemical Journeys podcast. Today I'm speaking with Gabriel Sigmund. Gabriel is an assistant professor at Wageningen University and Research in the Netherlands. His work focuses on the fate of chemicals in the environment and in man-made infrastructure, including their degradation, sorption and mobility. He's also engaged at the Science Policy Interface and is a board member of the International Panel on Chemical Pollution, an independent assembly of academic scientists contributing to shaping chemicals and waste management policies around the world. Gabriel, thanks for joining me.
Thanks for inviting me, Chris. I'm happy to be here.
I'm really happy to have you on the podcast. I've been really looking forward to speaking with you because we've had quite a few interactions recently through CTAC, the Society of Environmental Toxicology and Chemistry. Most notably, we've collaborated in establishing a new interest group for persistent science, and we've worked on some activities around that, and hopefully we'll get on to talking about that. I've also noticed that you've been quite involved in some really interesting publications recently, so it'd be great to talk with you about that as well. So before we dive into all of that, perhaps let's just start with you. Can you tell me a bit about your background? How did you get into doing what you're doing today, and how did you get to where you are today?
Yeah, I was born many moons ago in the northern parts of Italy, in the mountains of Südtirol, and I moved to Vienna to study there. And somehow I got stuck. I did my bachelor's and master's and PhD all in Vienna on environmental science, environmental chemistry topics. My PhD was surrounding sustainable strategies for soil remediation. Between my master's and my PhD, I also worked in a remediation consultancy, so I got a bit of insights into the remediation sector there. And after my PhD in Vienna on mainly soil amendments for remediating residually contaminated soils. So once you excavate the hotspot out, you very often have those soils around. It would be too expensive to treat in some aggressive manner. So that was kind of one of the topics I was looking at during my PhD. So after that, I moved to Munich, to the Technical University of Munich for a postdoc, working a bit more on transformation of organic micropollutants. I did not stay there for long for private reasons mainly. And after that, did a postdoc in Zurich, Agroscope and EAWAG, where, among other things, we looked at using sustainable activated carbon alternatives, so wood-based activated carbon, produced from waste wood, for removing mainly pharmaceuticals from wastewater effluent. And I moved back to Vienna after that. I got a junior group leader position at the University of Vienna, and there I started to work on a range of topics a bit more broadly. Ultimately, I ended up here in Wageningen 2 years ago, more or less, with the tenure track position that I have now. Throughout this whole history, so to say, more than a decade by now, one of the key topics or issues was always how contaminants, especially organic contaminants, how they behave in different systems, and more precisely, how badly or well they fix the stuff. That's really one of the key things that I have always been interested in. So that's what we call sorption processes. And this question of distribution, mainly between liquid and solids, has been around since the 70s, basically. In the recent decades, we have been confronted with a very wide number of quite different types of chemicals. So historically, we have been interested in those POPs, like your PCBs and PAHs and kind of the founding fathers of the Stockholm Convention. let's say. And those types of chemicals all had in common that they were quite hydrophobic and they really liked to stick to stuff. And one of the driving reasons why they stick to stuff is that they don't like water. That's what we call hydrophobicity. And those, let's say, newer types of contaminants that we're getting concerned about, like PFAS, like pharmaceuticals, and a number of pesticides and their transformation products, they all tend to be much less hydrophobic. In contrast to those, let's say, legacy pollutants, they very often have or can have a charge. So depending on the pH in the environment, they can ionize. And once there's a charge on the molecule, the behavior completely changes. So even though we had quite reliable, decently good concepts and models to describe the behavior of organic pollutants in the environment, basically in the 80s, those don't fully translate to this new generation of contaminants. And most of those concepts from the 80s are basically still being used by the majority of scientists, regulators, and so on. So a very primitive example, when we talk about sorption, this is the distribution between a solid and a liquid.
Mm.
If we want to compare sorption among different soils, you would normalize this sorption, the KD value, for the organic carbon content of those soils, and then you get a KOC value. So this is kind of the value, the universal value for sorption affinity in soil. And because it's not always easy to get those experimental values, again, back in the late 80s or mid 80s, I think people started with identifying relationships between the KOC and the hydrophobicity. And this is often quantified as the distribution between oxanol and water, so the KOW value. And this relationship to predict KOC works decently well, again, for those legacy compounds, but conceptually it's nonsensical for ionizable compounds. And that's really this issue, this problem, and how to kind of overcome this issue has been one of the core interests of mine for the last decade or so, to kind of from all kinds of different angles.
Yes.
Yeah. Sorry, I've been talking for a long time.
Not at all. No, that's a really nice way to frame it. And as the guy that studies how things stick to things, I think that's quite a nice way to frame it as well. And you're absolutely right. We are starting to turn our attention towards different types of chemistries than perhaps some of these legacy chemistries, the hydrophobic organic contaminants. You wrote a really nice paper along with quite a few others looking at sorption and mobility of ionizable compounds in ES&T, which I found it to be a really nice paper that laid out the kind of theory and the background to this topic, and then presented some really interesting conceptual frameworks and data for how we can tackle that new problem. Perhaps while we're on that vein, do you want to talk a little bit about some of the reasons why we're turning towards these different chemistries, and also if you want to go into any more about why ionizables behave differently, that could also be helpful.
Yeah, so from a purely academic point of view, what's really interesting about ionizables is that they can just have many different interactions with surfaces. So if we think about soils or sediments, they are basically almost always negatively charged. So they all have a negative charge. There might be some partially positive charged pieces in there, like some goethite maybe or something like that, some iron oxides. But considering that everything else is negatively charged, even if there is some goethite in there, it will be covered immediately by all the other stuff. So we can kind of assume it's all negatively charged. So on a very kind of basic primitive level, if we have a molecule that also has a negative charge, it would be repulsed. That's kind of very intuitive, right? And if we have a molecule that's positively charged, it would be attracted. Now, if we think about hydrophobicity, this KOW thing that I mentioned earlier, a charged compound always has a much lower hydrophobicity than the equivalent if it's neutral. So if we use hydrophobicity to predict sorption, we would get possibly a decently okay trend for acids that become negatively charged at higher pHs. But if we think about bases that become positively charged at lower pHs, those would stick more when the hydrophobicity is lower. So this concept really conceptually does not work anymore. And then on
Yes.
top of that, what makes our life more interesting and complicated is that if you have charges involved, a lot of other factors in there can also play a role. For example, if you have calcium in solution that has a divalent positive charge, so two positive charges, that calcium can bridge the negative charge with the surface negative charge. So you have all kinds of water matrix effects that you could more or less ignore for neutral compounds that now do play a role. So that's from a purely academic point of view, that's what is exciting and interesting about it.
Opened a
But
huge
from a
world of
more,
complexity. Yeah.
it's finally something to play because the world is too simple as it is. From a more, let's say, societal and implications point of view, or also environmental point of view, what this means is that, especially for the negatively charged compounds, those compounds tend to be much more mobile in the environment, which means they can spread much more quickly, much more widely. They can reach groundwater resources, all kinds of drinking water resources much more easily. And that is, of course, of concern. Quite recently, there has been this new substance class of persistent mobile and toxic chemicals that was introduced at the European level, at least. Kind of discussions going on also at higher levels, but for now, let's say at the European level, we have this new hazard class of persistent mobile and toxic chemicals and very persistent, very mobile chemicals. And if we look at those, the majority of those substances are ionizable or ionic. And that is precisely for this reason that mobility tends to increase if you have a charge involved, especially if that charge is negative. So there is a very direct implication for protecting water resources, for protecting the environment. Because if we go back to those legacy contaminants, if you had like a contaminant plume, let's say at an oil field site or whatever, that plume would spread quite slowly in the groundwater. And if you compare this nowadays with a PFAS plume, those spread super fast, especially if they are short chain PFAS. That's not a plume that you can easily contain anymore. It just goes everywhere really quickly, which completely changes how we need to deal with this issue of remediation, but also in terms of chemical management and risk assessment and all of those issues surrounding how we should or should not use chemicals is quite different for those kinds of substances.
Yeah, it's a good point. The issue of containment sometimes gets a little bit lost in the discussions about criteria. There is less of an opportunity to contain and remediate something that's escaping very quickly from a point source. Yeah.
You could extrapolate even further, right? If you think about the water cycle and if you think about very persistent chemicals, once they are out there, the extreme example, of course, is TFA.
Yeah.
We have TFA in the water cycle. It's in the rainwater everywhere. It's not degrading and it's not sticking anywhere. Some have claimed or have proposed this is basically beyond what we can call a planetary boundary or a system boundary. I would agree with this assessment because if something is not reversible, I would say this is some sort of a boundary that you cross. And I would argue the presence of TFA in the water cycle is not reversible. Of course, we can remove it with reverse osmosis or here and there, maybe with an ion exchange-based technologies, but those are very costly, very small-scale technologies. There is no way we are getting all of the TFA out of the water cycle.
Yes. Yeah, TFA. So that's trifluoroacetic acid. And
Indeed.
I don't know, Gabriel, do you want to go into that topic now, or shall we meander again?
What shall we do?
I think it warrants a discussion in it of its own, doesn't it?
Yeah.
But maybe we could keep on the topic of ionizables. So a couple of things I was going to ask you actually. So because one thing that I looked into quite a bit when I was looking at PAHs was how their sorption impacts their bioavailability, and as a result of that also affects other processes like their rate of degradation in different situations. And one thing I came across, which was quite interesting to me, was that this issue of bioavailability is it's not a fixed property, right? It kind of, you get this interesting phenomenon where the longer that a contaminant is present in a certain matrix, the more stuck it becomes, or the less readily it dissolves from that matrix. And that seems to be, or the theory is that these molecules are kind of infiltrating deeper into the matrix and getting more and more stuck over time and less able to be released. And just talking with you now about the issue of charged species, it seems a lot more related to surface interactions than sort of that kind of dissolving in,
Yeah,
say, the soft organic carbon component of a matrix.
the
Just off the
rubbery.
top of
Yeah.
your head, I'm putting you on the spot, really, but do you think that these age adsorption processes could be different also for these kinds of compounds?
Yes and no.
Mm-hmm.
So, of course, I did not want to completely escalate our very first question on the sorption interactions. But in addition to charge, a bunch of other molecular features also play a role, of course. And say, for instance, if we have some charcoal in a system, some black carbon or some
Mm,
activated carbon, a really important feature for a chemical to have or not to have is an aromatic ring. And many, many, many micropollutants do have aromatic rings. And those aromatic rings can interact with this carbon, some called glassy
mm,
carbon fraction, or the harder part of the carbon, in so-called pi-pi electron donor acceptor interactions. And, well, this is a very smart way of saying you have those delocalized electrons in the aromatic ring of your molecule, and as the universe loves chaos, they will kind of always push to the edges. So if you think about carbon on a molecular scale, that's activated carbon, for instance, that's a bunch of aromatic rings clustered together. So then you have this big cluster of aromatic rings. And again, the universe loves chaos. So all of the electrons will kind of push to the outer edge of this cluster. And then in the middle, in the center of this cluster, you have, like, let's say, a slight deficiency of pi electrons. So that's why those black carbon materials are considered pi electron acceptors, and the most micropollutants are pi electron donors. So that then means that basically those aromatic rings stack on top of each other and can really interact quite strongly. And for those kinds of interactions, I would imagine that we would see somewhat similar trends in terms of non-extractable residues over time and non-accessible fractions over time. If
mm,
we have this system, for example, with a black carbon or some kind of very humic carbon that has lots of aromatic structures, this kind of stuff.
mm-hmm.
If we had a different type of molecule, and I guess this is a recurring theme from now on, PFA, this
Yeah.
smallest, shortest chain PFAS, which does not have an aromatic ring, is negatively charged. It's very small.
Mm,
This one does not stick anyway, but let's pretend it would stick. It would not be as much of a issue, I think.
mm-hmm.
I would not dare to make a general
Yes.
call on this. I think it really depends a bit on the soil, or on the solid matrix and on the molecular features.
Yes. Yeah, I mean, that's it. It's such a crazy complex environment, right? And we're working at the micro scale, even the nano scale with these processes that are going on. So, yeah, you really have to adopt different conceptual frameworks to try and even envisage what is going on, right? And then I guess design
Yeah,
experiments to try and validate
indeed.
what's being discussed there. Yeah. So
Yeah, I
nice.
think I fully agree. And I think something that, especially nowadays in the age of AI and other types of energy sucking mega machines, there is a tendency to disregard the value of laboratory experiments and of
Mm,
really, just primitive sounding things. Put the compound into a mixture, shake it for a bit, leave it there for a while, and see what happens. But those kind of data are really essential for us to understand things, to confirm that what we think happens actually does happen. And if we now go back to this PMT substance class,
Mm.
There is an immense number of molecules that are already registered under REACH that are on the market that may or may not be PMTs,
Mm.
where inadequate, bad models that are kind of being stretched in terms of their scope and applicability domain are being used to somewhat estimate if those are or are not persistent and are or are not mobile. And at the end of the day, you, you probably really need to go back to the lab for many of those compounds and actually measure stuff. I think
Yes.
that's really, and the same thing now, if we now talk about this non-extractable residues, or we can switch this around in terms of bioaccessible fraction, there are experimental approaches to try and test this, right? We can incubate stuff for a while and then use an infinite sink approach, like a contaminant trap, to
Mm.
actually try to suck out as much as we can from the soil. And then we can basically, based on a mass balance, say how much can we get off or not.
Yes.
And I think that's, even though we know a lot about surface chemistry and environmental chemistry and how organic molecules do or do not diffuse, and we know a lot,
Yes.
but most of what we assume on those molecular scales, there is a degree of uncertainty and speculation there. And at the end of the day, to have some really hard data is, I would say, very often preferable. And
Yes.
I mingled in the past a bit, and that's kind of one of my hobbies, to be honest, try and develop kind of predictive approaches for, yeah, well, mainly for sorption, until now less so for persistence, but that may change in the future. And really, the limiting factor is not necessarily our understanding of the process. The limiting factor is to get good training data.
Mm.
If we don't
Yes.
have good data to train our model on, our model will not be good, no matter how smart we are.
Yes. Yes. No, no, you're absolutely right. I definitely see that a lot in the area of persistence and predicting degradation. I mean, a lot of the tools that we have in this area are still very basic. You know, they're trained on biodegradation screening tests, which have a very specific set of conditions that is poorly transferable to the real environment. Some of the models are even based on expert judgment, surveys of expert judgment. We do have overall a lack of data for degradation in real world conditions. We also have a challenge in that, which is very pervasive in the persistence world, of lab to field
Yeah.
extrapolation and relevance, because we take a sample of something from the environment, or we take an inoculum from a water treatment plant, and then we put it in the lab and we incubate it for a number of days, and we hope that what goes on in that vessel is exactly mirroring what's going on in the environment, which of course
Yeah.
it's often not. But very, very challenging and very costly to try to design an experiment that does better represent real world conditions. And you know, there are some good examples of that in the literature, but they are quite scarce.
I agree. I think I fully agree. And I do think there is really a need for those types of experiments. At the same time, even those very primitive, readily biodegradable tests, or what have you, if we had somewhat consistent data quality, and if we had enough samples that are being measured by different people for different chemicals, we could already take a much larger, stronger, further step forward
Mm-hmm.
than where we are now. Because I really think that this is a limiting factor. And I know we have more mobility data than we have persistence data in the PMP suspect list,
Mm.
which is quite depressing because the mobility data are the ones that I know quite well. And I'm very pessimistic about using those data to build really good models right now. So
Mm.
if I translate this into persistence, that means my hopes are even lower.
Yeah.
So that means really we shouldn't, I'm repeating myself now, but I really think before we completely give into this hype of having a big smart black box with some neural network or a random forest or whatever your approach may be. We need enough data to actually do something like that. And even if we have enough data, the parameterization of such models is still something we as experts must drive.
Mm-hmm.
So I recently reviewed a paper where it was clear that those people were machine learning experts, but did not really have a lot of domain knowledge. So they would
Yes.
just take correlated parameters and throw them all into one bucket and see what comes out. And something will come out, something always comes out. That does not mean this is something we should build on.
Yes. Yes. Oh yeah. Put much trust in. You're
Yeah.
absolutely right. It's garbage in, garbage out a lot of the time, isn't it? And
Mm.
I would say that there's been some good progress in the last 10 years or so on data curation, manual data curation. Coming up with bases to evaluate the quality of the information and its reliability and relevance. So a lot of work on aquatic toxicity data, thinking, the CRED scheme. John Arnott and colleagues have developed a similar system for bioaccumulation data. And then I was lucky to be able to work on the topic of persistence, and we have a persistence assessment tool which does similar things. So, helps you to actually look at a study and understand, record, and then evaluate different aspects of the study and how they may impact the quality of the study. And a manuscript due to be submitted on that. So people
We all
can
look forward to
look
reading
out
it.
for it. Yeah, no, great. But yeah, the
Okay.
PMT concept is really interesting as well for me from a policy and an implications point of view. I like looking at all these policy developments, also trying to think, well, what does it mean in terms of implications? And I think you've highlighted that we do have a lack of data to actually do these assessments right now. But the first thing I would say is that creates a big challenge, because it's now a requirement. I think we're in late April, so as of the 1st of May, all substances placed on the market in the EU should be classified according to these new hazard criteria, the PMT and the vPvM. And there's a situation there in that you have a lack of data. So I'm not expecting to wake up on the 1st of May and find that all these dossiers are going to have up-to-date PMT classifications suddenly
Wouldn't that
appear.
be nice?
So I don't know what I don't, but it does
Yeah.
kind of puzzle me, because
Yeah.
I don't know what the regulators are going to do about that, because in a way there's going to be a lot of question marks over
Yeah.
everything. And in the past, at least from an environmental point of view, it was possible and it was expected that organizations that placed substances on the market would classify them according to their hazards, and it was generally possible for organizations to do a reasonable job of classifying the environmental hazards based on the schemes that were in place. We're entering a world now where there's going to be big gaps in these sections for the hazard classification, and I wonder whether that's going to change the way that the whole CLP regulation really works. Is it going to take the onus away from the industry to classify and over to the regulators to take more initiative in driving home some of these more complex hazard classes?
That's a really interesting point you're making. Well, I think there are two different issues here. One is, let's say, for new chemicals that do enter the market that have not been registered yet. And there, I would say, at least from my point of view, the burden is not that excessively heavy for you to include those two experiments, let's say. As well, of course, depends on what volumes you're producing as a
Mm,
chemical manufacturer or whatnot. But I would say for new chemicals that enter the market, I, I would be cautiously confident that this is perfectly feasible, as it was feasible for many other hazard classes before. Of course, looking backward, for all the chemicals that already are on the market, for all the stuff that already is there, and let's be honest, that's way more than will
mm,
enter in the next 2 or 3 years.
mm-hmm,
And
mm-hmm.
that's a tricky one, because
Yeah.
I doubt that we would even have all the commercial labs in Europe would even have the capacity to produce those data in the next couple of years. I
Oh
really
yeah,
doubt that. I don't think that would be possible.
yeah.
So
There's no
indeed,
chance. Yeah.
what may or may not make sense, this is just from the top of my head, is to have some kind of a prioritization on which dossiers need to fill their gaps first. And the obvious one is production volume, but after that, you may still base a prioritization on some of those tools that are semi-reliable that we mentioned before.
Mm.
You'll have to do something, and we'll have to use the best available technology there is. And well, I guess a screening based on DOW and charge state, for
Yes.
example, in terms of mobility, and maybe using one or two of the predictive tools for persistent that are out there. That might
Yes.
be a first step to prioritize. Yeah.
Yes. Yeah. Prioritization is really important here, and that is one thing that I detect is that what we have done now, having had a looked at the screening data for biodegradation over the years, I would say that roughly a third of the chemicals that are on the market meet the ready biodegradability criteria and therefore are on the off ramp for this situation. But everything else that's an
Oh,
organic
we're good then.
chemical,
It's just
yeah,
2/3.
yeah, you've got the other 2/3, and
Yeah.
I see no other off ramp
Yeah.
than doing the simulation tests. Whilst there are other screening methods of predictive tools, and you can build what's called a weight of evidence, the burden of proof that regulators would finally accept is too high with the available techniques that we have. There isn't really anything that can adequately replace the results of a simulation test, at least under the EU REACH framework. We also have the issue of transformation products. What happens when chemicals are being degraded? They're being transformed into other things that are becoming more polar and likely more towards the mobility criteria than the parent substance, if they haven't already exceeded the threshold. And therefore, the question then becomes, well, what is the persistence of those transformation products? And if any relevant transformation product is found to exceed the persistence threshold, then the assessment goes right back up to the top and assigns that criteria to the parent substance. So, when I think about this, I find it very challenging because I think that, everything is suspect, and without simulation test data, we have few tools available to robustly answer the question of, do these chemicals fulfill the criteria? And as a result, I think we're going to be having a lot of uncertainty for a long time,
Yeah.
potentially.
Yeah, I hear you. I agree. But I think there are also different levels to this issue. And let's say on a more high, big level thinking side of things, there is this concept of chemical simplification that
Mm.
right now we have something in the order of more than 350,000 man-made chemicals out there. And of course, for the overwhelming majority of those 350,000 chemicals, we know close to nothing in terms of their environmental behavior and so on. Well, most likely, we don't necessarily need to have 300,000 distinct chemicals out there. Most likely, many of those were produced for a number of reasons, which might be linked to IP and patenting and renewing market approval because this different side chain makes it a new molecule, or well, all of those reasons better than me probably. But I do think that all of the issues and reasons you just listed are actually a very good motivator to say, well, maybe, as a society, let's take a step back and think about how many pieces of the chemical puzzle do we really need? Most likely it's less than 300,000. And
Mm.
that would already kind of bring us forward. If we say, okay, those are really the pieces of the puzzle that are really essential, that we need to build all of our different functional chemicals that are essential for human society to work. I think that is, of course, living in the world we live in. This is not going to happen by the 1st of
Yeah.
May, at least not this year. But I think those are thoughts and considerations that we should keep in mind when debating such issues, because to some degree, this problem is self-made. Why do we have so many different chemicals? Why do we need so many distinct chemicals? Do we really need so many distinct chemicals? There are smarter
Mm.
people than myself that argue no, and I would tend to
Yeah.
agree. On the more day-to-day side of things, I do think developing not just computational tools, but also developing experimental screening tools is really something that can also make a difference here.
Just on the topic of simplification, because I have heard that discussed before, and yeah, it's an interesting idea for sure. I think you're right in that the way that the chemical market has originated through innovation and competition has driven large numbers of chemicals to be invented and develop markets for them. I guess, I hear the 300,000 number for chemicals, in inventories and whatnot. I guess we do have some good knowledge, at least in the EU, of that it's a smaller number of chemicals that are being used in large volumes. And the other thing I would say is that the PMT vPvM kind of process is going to affect, I think, classes of chemicals probably more than kind of like individual chemicals.
Mm.
So, but that's not to say that it's not a valid thing to consider.
Yeah.
Yeah, whether the mechanisms are there to to
No,
implement
that's
something like that is another matter.
the argument you're making holds true not just for the PMT class, but more or
Mm,
less for any class. You could
mm,
make the same argument for PBT, and you could make the same argument for this proposal of PFAS as a class.
yeah.
Those are, of course, very different types of classes and groups. But I would argue that it's a healthy thing to switch the assumption around. You're not innocent until proven guilty. You're guilty until proven innocent. And if you are not innocent, you can still be essential.
Mm,
So if we think about the PFAS restriction or potential PFAS restriction as a class, as a group, there has been a lot of voices claiming that this is threatening the energy transition. And we have
mm,
many pharmaceuticals that have a fluorinated side chains. Well, pharmaceuticals are anyway not regulated in REACH. That's a different story. But there have been a bunch, and fluoropolymers for medical devices and whatnot. So there have been a bunch of uh, more or less loud voices
mm-hmm.
that made a case against PFAS as a group with some very specific use cases that may or may not be essential. I would argue many of those voices are right, but the reasons they list are not a reason not to regulate this as a class. They're just a reason why some of those chemicals may still have an essential use until we find a better solution. And I think that's how I would also approach the PMT and any other hazard class. Okay, so your chemical is super important. Good to know. If you show me the receipts that it is actually essential, well, then most likely
Mm,
we will find a way to keep using it until we find something better. And I
mm,
think that's, from my point of view, that's the right incentive and the right pressure to incentivize safer, better chemicals on the market than to question the whole group. I think there's a reason why we have those criteria and why PMT as a class has been proposed. Those
yeah.
features are a profile of a chemical that is threatening to, as we discussed earlier, to water resources, to the environment. They spread more widely, they stick around for longer in terms of time, not in terms of absorption. um, there's a reason why PMT as a substance class, I think, should be regulated. And if some of those PMTs are essential or important or societally like super valuable, great. Let's discuss this on a case by case basis. And let's say, okay, if we do need you, let's keep you until we find something better.
Yeah, it's a fair perspective. because I get sort of down in the weeds of assessing chemicals, looking at data and things and seeing how some of the tests work and some of their deficiencies, let's say. That's where I become more troubled by it, because I think a lot of the concern that's poured fire on the issue that produced the PMT hazard classes was for highly persistent substances like certain PFAS chemicals that have been identified to be a real societal problem. But then when I see that there's a test where the results are really noisy, and on one day you'll get it as VP and another day you'll get it as not P, and it's a complete lottery, down at that 40-day half-life threshold, that's when I'm kind of like, ooh, because we implemented this because everyone was talking about persistence with a PFAS in their mind, and now we're looking at things that degrade, somewhat slowly, but they're not the same kind of planetary boundary threat as some of these other chemicals.
No, I very much agree. I think we really have to distinguish the PMT and even the vPvM. Those are threatening, as I said, the groundwater in terms of how mobile they are, in terms of how long they can persist. That is a threat. But what I think you and I would call extreme persistence, that's a different story. And that's something where I also am way less flexible about, stick around and let's see what happens. Like if you are extremely persistent, I would argue then you should not be around. Like a fully fluorinated carbon chain, no matter how long or short it is, that basically does not degrade in the environment, should just not be there. And if you have a super duper essential use, you better hurry to find an alternative to replace that. Because that's a different story. Like if something degrades in a year or two, that's not nice, but it will degrade in a year or two. But if something just does not degrade or degrades in 50 years or 100 years, that's a different game. So with that I think bridges us nicely to the TFA story.
Go.
Because I think it cycles back to some of the things we discussed earlier. So indeed, TFA is for those listeners who don't know, this is, let's say, the shortest PFAS out there. So it's basically just one fully fluorinated carbon and then a carboxylic acid attached to it. So it's kind of a fully oxidized something, and this is what remains. So we can assume for most molecules that have a carbon, let's say a carbon with 3 fluorines attached to it as a side chain somewhere in the molecule, eventually it will oxidize to TFA. There are some exemptions, and there are some special cases where you have some heteroatoms in the chain length, and well, there are some issues there, there are some things that are different, but in the overwhelming majority of the cases, this will be the final product if your molecule oxidizes. TFA, as I mentioned earlier, is very small, is negatively charged, does not stick anywhere, does not degrade. We have some evidence now that it also has some toxic properties and features. I'm no toxicologist, so I don't want to comment on this too much. We have some evidence, that's all we know. But even if we had no evidence, I would argue the fact that this thing goes everywhere and does never break down, we should just avoid it. Why would we take the risk? TFA is known, or one of the main sources for TFA is known to come from the atmosphere. So we have those F gases that are used as cooling agents and other industrial applications. And those F gases, let's say, form a majority of the TFA that we find all over the world, that by now form like a baseline of TFA that we can even find in rainwater and so on. And that is already worrisome and this planetary boundary that we mentioned earlier. So last year, together with Hannah Jös and Finjan Freling, we and a bunch of other co-authors, collaborators, we collected data on pesticides from all across Europe and also from the US. And we also tried to get some data for China. That was unfortunately a bit more difficult than for the other two. But we collected those data looking at pesticides that have, let's say, TFA precursor side chains, so they can potentially form TFA. And we use those data to make estimations about how much TFA could be formed on a spatial scale. One big problem with those estimations, or with any estimation in this direction, is that we have little to no data on actual TFA formation rates, because this was never a requirement for registration until now. And the fact that pesticides are TFA precursors until now is still not a reason for them to not be approved for use in any of those regions. So we actually made two different assumptions. One was kind of a very conservative one, that 100% on a molar basis will become TFA. That's probably a bit too pessimistic, but still, that was one assumption. The other assumption was based on some conversations that we had with industry representatives that said maybe something like 30%, which is probably a bit optimistic, I'm guessing, because of the source that we got it from. But basically, we kind of looked at both of those scenarios. And for both scenarios, we actually got, even for the lower, so to say, we got, to me, quite impressive numbers in terms of how much TFA could be formed and thereby released on an annual basis because of agricultural practices, basically, with those pesticides. And then we were very lucky to find groundwater monitoring data from a region in Germany from about 1,500 wells or so, where TFA was monitored or measured. And we linked those concentrations to the land use categories above ground, which is also not as obvious and easy as it sounds, because land uses are not always as clear and not as well documented and reported. And then if you think about groundwater, there is a certain flow direction, and there is a bunch of stuff that actually complicates things. But even though we ignored all of those complicating factors, we could still see a quite significant increase in TFA concentrations in agriculturally affected aquifers compared to the ones that were not. And that is a quite strong indication that pesticides are a substantial source for TFA. And in some European countries, like also in the Netherlands, TFA is already a regulated substance in terms of drinking water regulation. So this can really become something of concern in terms of drinking water safety. And from a scientific basis, I would argue TFA is a PFAS. TFA is extremely persistent. There is no question that we should not have it in our waters. We know it is there to stay. We will not get rid of it anytime soon because it's everywhere. Pesticide use, I would argue, is really a source that would be something that could be we could take action. It's not something that is impossible to get a hold of. So I'm cautiously optimistic that this debate has this paper, among another paper by Hans-Peter Arp and colleagues, who kind of highlighted TFA as a planetary boundary also last year. I think together with that paper and some other debates and people raising their voice, I think the awareness of this issue there is awareness for this issue now, let's put it this way. The week that we're recording this, the PAN network, this is a Pesticide Action Network, an NGO, they monitor TFA in a relatively large number of wine samples of different vintages, and they've found really shockingly high TFA concentrations in those wine samples, like some of them were several hundreds of micrograms per liter, which is really, so that thresholds for PFAS are in the lower nanogram per liter range. So this is really a crazy amount that was found in those wines. And some of those CFA precursor pesticides that we looked at were fungicides. And I think that wine production is one area where fungicides are quite popular, let's say. So I could imagine that that is part of the story there. Yeah.
Mm. Okay. Yeah. I have to say on this TFA topic, I mean, although it seems to have, I mean, people have obviously been talking about it for a while, but it really has sort of kind of exploded onto the scene. And the time when I, first really took note was at the last CTAC conference in Seville, seeing the presentation from Fidian Freeling showing the dramatic increase in the concentration of TFA that was being found in, I think he had historical plant samples, right? And plants take TFA up very effectively. He seemed to demonstrate that the concentrations in these plant samples had gone up
it's scary.
fourfold between 2010 and 2020, which was, of course, quite a startling thing. And you're right in that the concentrations are generally much higher than most other PFAS and can get to appreciably high levels already in surface waters and things. And I wonder if you agree that one of the disagreements comes from a difference in worldviews, perhaps, in terms of that some people kind of, as you said, this chemical is here, it's changing the composition of the biosphere and whatnot. So, and we're doing it and it's not going away. Therefore, it shouldn't be there. And then others kind of take a more laissez-faire approach, let's say, to that. There are byproducts of human activity, and we're generally comfortable with that fact. If we're confident that they're not causing adverse effects, and yeah, that is my sense. But it certainly sounds like like you said, the momentum is really there now on TFA, and I think this also fits with the sustainability, the global sustainability agenda has kind of caused a shift in worldviews for a lot of people and the general public as well.
To comment on those two views that you mentioned, I mean, I made my position clear earlier anyway, but I think from a very fundamental point of view, we could argue it's a difference between historically contamination and pollution. I don't know if you read the book Pollution is Colonialism. I highly recommend this book. It's a book by a North American researcher. Their name is Max Liboiron. I don't know how to pronounce their name correctly, but and what they argue is that colonialism is about taking someone else's space or taking something. The origin story of the term pollution comes from contaminating rivers, surface waters. A river was considered polluted once the capacity of the river to take up the contaminant was exceeded. So anything below that was just contamination. And once we are disturbing the ecosystem, then it's pollution. But the fact that as humanity, we assume that we have the right to put something in there that was not there before, that's what this book basically argues. That assumption is colonial because it assumes this is ours to change, this is ours to take, this is ours to modify. You could even go back to Christianity and, we are the rulers of the world and all of those anthropocentric ideologies around our relationship with nature. And I think it really comes down to how do we as humanity relate to our environment, to what is around us. Are we part of it? Are we estranged from it? Are we its rulers? Do we have a responsibility towards it? That's a very moral and a very philosophical question. So I highly recommend that book. I think it's a good read.
Yes.
But apart from that, I think that's what those two, apart from, of course, having different interests and people wanting to make a living, and that's all fair. But apart from those different interests that may surround those positions, I think it's really about the question, how do we as humanity relate to our environment? What are we responsible for or not?
Yes. 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 No, no, I'm really happy that you brought that up because I see that every day, kind of working in this topic is, yeah, we're scientists, we're natural scientists, we're studying physical processes and biology and whatnot. But actually, there's so much cross-cutting, philosophical, sociological, deeper, meaningful questions that we really grapple with, and that the work that we do really depends on. So you've drawn a bright line between a duality there in terms of different people and how they see the world, and therefore how they will interpret all of the phenomena that we're studying. So that's really good. Good. So and perhaps we can if you're happy to, we can talk a little bit about the persistence interest group.
Yes, yes, it was on my
Yeah.
wish list here on my looking down at my wish list. Because
Yeah.
I also think, because that's actually, I think it's a good transition. Well, I don't think we have any uh, evil doing destroying egomaniacs in the persistent science interest group, but
I hope.
no but what I really appreciate about this interest group within CTEC, so this is that, well, you are chairing and I'm a part of the steering committee, um, is that we really also bring different views and different sectors together, because that is the nature of CTEC, that there is this philosophy that you want to bring academia and industry and regulators to kind of talk and discuss and find common ground and ideally work on making the world a little bit less horrible, maybe. Um, that's maybe a bit overambitious, but the fact that um, CTEC is the way it is, I think makes it a very valuable forum. There are things that I do in my academic life or also in my private life where I would maybe rather stick only to academics, and there might be things where an industry person would only want to stick with industry. But I think CTEC is a really valuable place where you meet people, where you confront each other with your different positions, and that's what makes CTEC really exciting and valuable. And I think that's what makes, among other things, this interest group really exciting, because all of the steering committee members, I believe, are deeply knowledgeable, have very unique points of view and also positions that might not always align perfectly, but I think it's very valuable to learn from each other and to exchange those positions, because that's how we can grow. If we stick to our own microcosm, we won't. We will just, well, reproduce
Yes.
what we already believe and think. And there is a really, I would argue, a really deep well of knowledge on persistence and issues surrounding persistence in the persistent science interest group. So if you're listening to this before a CTEC meeting, chances are that there will be a persistent science interest group meeting, and you should go there and meet some exciting, cool, and interesting people. And the other thing about the persistent science interest group is that I think, if we go back to this question about hazard classes of chemicals, like sometime earlier in our conversation, I mentioned PBT, PMTs, and PFAS. What is the feature that connects all of them? Well, it's persistence. Persistence is the molecular feature, and I think you have recently also written something about this issue of persistence, right? And how this is like the key feature. And this is really something, especially if you're a young academic that wonders, okay, what is a subject or a topic where lots remains explored and whatnot. I mean, persistence, we all have a rough idea on what it is, but persistence is really I cannot imagine a molecular feature linked to persistence that would make a chemical inherently awesome and safe. If a chemical is persistent, my default setting is always to be concerned. So to understand and tackle persistence as a chemical property is really key if we want to develop safer chemicals, if we want to regulate hazardous and dangerous chemicals, if we want to do anything at all that has a positive impact relating to anthropogenic entities and the environment. Understanding persistence is key. And the persistent science interest group is one place where you can learn about those issues. Yeah.
Yeah, no, definitely. It does, puzzle me, that persistence seems to have been the unwanted kind of cousin of ecotoxicology and even other fate processes sometimes, but it definitely feels like it's having its heyday now in terms of the interest. And I love the fact that it is such a rich and diverse and multidisciplinary topic that there are literally infinite ways to explore it, and we've touched on some of them on this call. The fact that it's integrating lots of other processes like sorption, bioavailability, and the complexity of the environment and how that influences whether chemicals will degrade or not and what they may degrade into. So yeah, it's so rich, and it's been a real pleasure collaborating with you and the rest of the steering committee on that, because, whilst we do have varying views and perspectives and interests and all of that, it has been a very positive, warm, friendly collaboration amongst everyone. So that is, I think, another real success of the group so far. Perhaps we could just talk about, so we mentioned earlier on that we did a webinar on extreme persistence that you and I collaborated on along with several others, and we had a very interesting panel discussion there that it's still available online for anybody that wants to stream it, so we'll put that in the show notes. You're also currently working on another webinar with Ina Vemberg, right? Do you want to just briefly mention that?
Yes, and that relates to one of those wonderful problems you mentioned in our call earlier today, that when we have some kind of an environmental inoculum and we want to derive a DT50 value or some kind of a persistence descriptor, what we tend to see is that wherever we take our inoculum from, however we store it, whatever we do with it, we always get exactly the same number. No, unfortunately, we do not. So Aina is really leading this effort, but well, I'm one of the people that tries to support her in this. But we're organizing this webinar really with the aim to look at what can we learn, how can we understand how the microbial activity and composition and kind of how the biological side of our biodegradation testing systems affects the outcome. And shockingly, in the preparation for this webinar, I learned from quite a few experts and some of the speakers that we will have. There is the standards in terms of describing the inocula for many of the reported DT50 values or persistence descriptors, it's quite low. So just like really basic things like I don't know, respiration rate or some kind of indicator for biological activity in the test system, those kind of things are already an important starting point to kind of some degree, benchmark or harmonize and see, okay, maybe inoculum was like way more active or more alive than this other one. That's where it starts. But of course, then one can really go into microbial composition and kind of what enzymes are there and all kinds of different aspects that make life interesting. But in a perfect universe, at the end of that webinar, you will understand a bit what factors about an inocula make it be more or less likely to degrade a compound, so to say, but also what are best practices in terms of experimental setup, but also in terms of reporting and in terms of, making your study or your biodegradation test, more comparable to others, because that is right now really something that the community is struggling with. Yeah.
Mm,
Yeah.
mm. Yeah, I mean, they talk about it as a black box in that, there's very little understanding of what goes on once you put your chemical into the vessel and how the microorganisms, what the microbial community is doing and the importance of that for degradation. So, yeah, congratulations to Ina and yourself on getting that webinar organised. I'm really looking forward to it, and I hope that we'll make some good progress there. And then it's just worth mentioning as well that we will be having a meeting at the upcoming conference in Vienna. So if you are attending the CTAC conference in Vienna, folks in the audience, then I really invite you to that meeting to come and hear about what's going on within the interest group. We've got a really exciting agenda with lots of different projects to discuss. It's important to mention that somebody who was very prominent in the persistence world, John Parsons, sadly passed away towards the end of last year. We've decided to dedicate the meeting to John, and we'll have a part of the meeting where various people will reflect on John's life and his accomplishments, including his wife Annika. So that promises to be a very special meeting indeed.
Yes, I could not have said it any better. Yes, at this point, it's almost awkward to mention that in addition to all the wonderful things in the persistent science interest group that Chris just mentioned, together with Hans-Peter Arp, we are guest editing a special issue or a team collection, as it's called, in the Royal Society of Chemistry journal, Environmental Science Process and Impacts. And that special issue will be about, or is about, transformation products and mixtures. So those are all of the really complicating factors when we want to describe chemicals. So we actually barely touched the issue of transformation products today. We could have probably talked another hour about that.
I know.
Because that's a really tricky one. In most regulatory frameworks globally, if at all, we're talking about parent compounds, and transformation products are completely ignored. And especially if we think about regrettable substitutions, if we think about all of the potential issues with developing chemicals and using chemicals that will be a headache down the road, transformation products are really a big one. So the TFA issue that we came back to a bunch of times, that's a transformation product issue, but that's just the tip of the iceberg. There are a bunch of transformation products out there that can be a real headache. Another example that was, well, by now I have to say a couple of years ago, researchers in US had found that fish were dying in a pond next to a road a couple of years back, and they really worked a long time to find out why the hell this was happening, and found out it was a transformation product from a tire additive. So there was this antioxidant that is added to tires to kind of keep them soft and rubbery and sticky for longer, I guess. It's called 6-PPDE, and the transformation product 6-PPD quinone is basically super toxic to fish. And well, this is one example that took really smart researchers a long time to find out. So who knows what the jungle of transformation product that is completely unregulated in the majority of the universe. Who knows what is down that rabbit hole? Yeah.
No, definitely. No, it's a really important topic, and yeah, congratulations to yourself and Hans-Peter on the special issue, and I really look forward to seeing what work is published in there.
Amazing.
Great. Well, Gabriel, this has been fantastic. We went on for longer than planned, so I appreciate you hanging on.
Cut out all the bad stuff.
Yeah, exactly. Yeah. So, but what I normally do to wrap up is I ask people 2 questions. So the first one is, what is a big goal that you're working towards in the coming months?
In the coming months?
Yeah, it's probably something you've already mentioned.
Yes. Now I need to. A big personal goal of mine is to be still motivated and to stick in the coming month, because a lot of meetings and a lot of traveling and a lot of invitations to give here and there contributions, in addition to chairing a bunch of sessions at CTAC, which I'm very excited about. But I'm also aware that at the end of that run, I will be exhausted. So my one goal in the next month is to not be exhausted at the end. And another one is relating to this extreme persistence paper that, or not paper, the webinar
Yes.
that you mentioned, but now this was a Freudian slip. We are hoping to write something up on that webinar, and that's something that is kind of high up on my wish list for the coming months. Yeah.
Yes, good. So we'll get some momentum on that hopefully in the next few weeks as well. But yeah, so thank you. Yeah, make sure you conserve your energy through these busy months. Yeah, that's going to be a challenge. And then the other thing is, one piece of advice that you'd give to somebody starting out in their career.
Yeah. On a content level, indeed, I think transformation products and persistence, and maybe the combination of those even, persistent transformation products, there is a lot to discover in that field. On a more personal level, more often than not, the people you work with are more important than the details you work on. So choose your mentors, your supervisors, the group you're working in wisely. It sounds so trivial, but it's really, really key. Yeah.
Yeah, no, it is really important. No, that's really good advice. A bit of practical, and then a bit of a bit of subject specific advice. So no, that's great. Thank you so much, Gabriel, and
Thank you,
thanks.
Chris.
No problem. And thanks very much, everybody else, for listening in. Your time's really precious, and so I'm really grateful that you've spent it with us. If you've enjoyed this podcast, then please tell people about it who you think would enjoy it, and I hope that we will continue to explore this story of chemicals in the environment with more people in the coming months. So more conversations like this to follow. Thanks very much again. Thank you, Gabriel, and see you in Vienna.
See you all in Vienna, hopefully.